So what I'm going to talk about, when I finally get there, there'll be a little bit of speculation along the way, is sort of a slightly long and rambling story. It's something we've been trying to unpick now for about 6 or 7 years. And you'll see actually a little bit of the progression of our thinking about it as we go along, because I've kind of basically tacked on to the end of the new slides as opposed to overhauling the whole thing. Anyway, so, and the idea, basically we're trying to sort of understand the origin of an observation that's an experimental observation, which is basically that if you shine a whole lot of light into the motor cortex in a primate where you've infected pyramidal cells, we think, with an excitatory opsin, we'll talk a little bit more about what that is. Basically, you inject a huge number of spikes into the system. You can see those spikes, there's a lot of extra firing, and then you turn the light off, it all goes away, and the system just keeps running. And there's something rather peculiar about that, because we think of these circuits that we're pouring the light into as actually driving, like the dynamical system within them, as being a large part of what drives the movement. And so we've been trying to understand whether we basically have to revise the way we think about what the motor cortex is and what it does, or whether there's some way to actually put these ideas together that you both have a dynamical system creating movement, and yet you can perturb it hugely and have that perturbation basically be quenched almost immediately. So that's where we're going. Let me start off by again acknowledging the people who've actually been kind of at the coalface working on this. So the data that I just kind of sketched, but we'll look at in a bit more detail, were collected by Dan O'Shea and Werapong Gu, working in Krishna Shenoy's laboratory. And then Werapong went off to start off his own lab. And since then, Dan and Krishna and then Leah Dunker, a graduate student with me at Gatsby, have sort of been staring at these data and trying to understand them. In fact, this was Leah's master's thesis many years ago, and she's coming to the end of her PhD at this point, and we're still kind of, she's done many interesting things along the way, other than what I'll talk about. But this one is sort of has still been making slow progress as we go along. Okay, but I wanted to start actually in the context of the workshop here, just sort of thinking a little bit about these sorts of dynamical systems and ideas about manifolds and why it is that we might find some kind of low dimensionality in neural data. And so I think many of us are comfortable with the idea that a lot of computations really take time within the brain. Through a lot of that time, there aren't sort of sustained external inputs arriving, and so it looks like basically, you know, we're watching thinking. We're watching some sort of recurrent process within the brain itself, either within single local circuits or perhaps between different local circuits, that is achieving the computation that may be expressed in a behavior. The components that go into that seem to be individually quite noisy. And when I start off by saying spikes are noisy here, I'm not pointing to the fact that we can repeat an experiment and get different spike trains each time. I'm pointing to the fact that we're fairly confident that what will actually happen in an individual synapse as an incoming spike comes in looks like a random process. It will release a vesicle or it will not. And the probabilities involved there are often, you know, not sort of in the region of 0.5, you know, less or greater, but in either case the entropy is high. There's been some calculations arguing that even things like thermal noise of opening and closing of channels in the membrane ends up being amplified near spike threshold and changes the timing of spikes. That's something that's very hard to measure directly, so these are calculations. That's true if the cells are sitting near the spike threshold, but when you go in and measure membrane potentials in animals, you often find that they are indeed sitting near a spike threshold. We know about branch failures, propagating action potentials in axons and in dendrites will fail at various branches. And we also have reasons to think that these sources of noise, which may or may not be small, are then embedded in a circuit with strong nonlinear amplification. And we should expect there to be some form in quotes perhaps, of chaotic amplification. Again, people have tried to make statements about each extra or misplaced spike will create 23 downstream spikes. Then obviously that's a very rapid exponential explosion in how quickly the system will become disordered. Okay, so now, you know, probably, but we don't know this for sure, some of this sort of low-level biophysical stuff, and maybe this what people sometimes refer to as microscopic chaos, does contribute to the variability that we see when we try to repeat an experiment as carefully as we can. But again, that's something that we don't know. We don't know what the origin of the variability in repeated experiments is. But nonetheless, and this is a slightly hard statement to make, but by some measures, comparing apples and oranges, it looks like we can achieve behavior much more reliably than the activity that we would measure in a single cell that we again presume is underlying that behavior in some way. And again, it's very hard to compare variability in two entirely different things. We've tried to do it by sort of likelihood distributions and things like that. But it sort of conforms with our intuition. We record from individual neurons, and on a single trial, you'd be hard pressed to actually say what was going on, but the behavior is fairly consistent. Okay, so somehow that noise in components, variability in individual neurons gets quenched sort of in the process of creating behavior. And so we think that that process lives somehow in the network. Yes?
I have a question about this last statement.
Yep.
Recent papers which show that when we measure high-dimensional behavior, which can explain way more variability in single neuron firing, but to what extent this last statement is restricted or limited by low-dimensional measures of behavior?
So you mean that there's something expressed in the behavior which...
Because the classical approach to quantify, let's say, what is the proportion of left versus right choices, and what is a hunter checkly in the arm movement task, right? While recent studies image the whole body movement of the animal and found that a lot of variability of single neuron responses can be attributed to task-irrelevant behavior.
Yes, so it's possible, I think. At the single neuronal level, I think there's still... So certainly in the primate work that we did looking at movement variability of the arm, it was only the arm. We didn't have measurements of anything else. So now, again, the story, at least, is that there's a topography of different effector control across the motor cortex in the primate. And so the cells we're recording from were in the arm movement areas. How much of that might be connected to something else, to postural control and things like that, we just don't know. Okay, so how do you build a network to avoid some of these things? Well, at least two possible ways of going about it, right? We can think of a sort of a digital solution, and this is really the idea that inspired, I think, Moshe Abelos's thinking about the SynPhi chains, is that if you can set things up so that at any moment the total input that comes into every neuron is either very, very high or very low, then even if there's some amount of variability in the precise timing of the incoming spikes, that will be reset and you'll get a much more reliable firing at this point. Okay, so noise processes some of this low-level, this kind of spike noise in this sense, so a failure of a spike, for example, would not necessarily disturb the firing of downstream cells. And this sort of led to people thinking about SynPhi chains, in Abelos's case, SynPhi braids or polychronous networks in the case of people like Eugene Izakevich. We've lost the bottom of this one. But another way to do it is sort of more closer to the sorts of things we've been exploring here, which we might think of as an analog solution. And that is to, in essence, create some sort of averaging process across the network as a whole, where the averaging is acting in some sense on low-dimensional, I guess we could call them latent variables. They're latent variables from the point of view of the experimenter looking at the system. But you might prefer to call them something like a computational state. Okay, so in this view, the computational state is something carried in the activity of many neurons, but it's lower dimensional than the network as a whole. And its dynamics may actually may or may not, turn out to be somewhat simpler than the complicated, chaotic, and nonlinear dynamics that individual neurons express. And so, even though the neurons are noisy, and the neurons we record are, modulo the question of whether we're recording all the behavioral activity, noisy, the noise, in effect, so in this picture, the noise would average out through the recurrence. And therefore, if we can record enough of these neurons, then we might be able to see this network state despite the variability in individual neurons. By looking for what's common, we can try to extract that computational state. Right? So this is the idea that many of us have been pursuing, sort of from our point of view, it sort of kicked off in a NIPS paper in 2006 when we were trying to think about dynamical systems that underlie this. And then there was this review that Mark and Krishna and Byron and I wrote, thinking about sort of systems that might work this way and how to analyze them. Okay, so one of the questions that sort of this might bring up is the question of whether having a dynamical system that achieves this sort of noise averaging through some kind of low dimensionality in the projection imply that the data we will measure from the system will be themselves low dimensional or not. And that's important because if we just use a dimensionality reduction technique to look for structure, we're looking for a manifold near which the data lie. And that makes sense if indeed the data are concentrated near a manifold. But it may be that a dynamical system that whether, say, the network connectivity is effectively low dimensional, so we're taking the high dimensional activity, squeezing it down into low dimensions before pushing it back out to affect all of the neurons. The activity moment by moment might happily lie off that manifold, okay? But it's only a projection onto the manifold of the data through the connectivity that actually dictates the future evolution of the system. Okay, so that's sort of the separation that I would like to explore to some degree. Okay, so let's start with this data set that we've been thinking about for a while. So these are data that were recorded from the motor cortices, both pre...
Okay, yeah, sorry. Can you just clarify with regards to your hypotheses?
Yep.
Or, this question between the two that you're trying
Yep.
to adjudicate. Do you know Gao's work in Sergei Guguli's lab, where they say, okay, if you have a low-dimensional behavior, then we, can make some simple assumptions on how that could be projected into a high-dimensional space.
So I don't think they're not very simple assumptions.
Okay,
so the problem that goes into that, well, maybe that's a offline discussion actually, just because it will take quite a long time. But I think part of what we're after here, though, is also thinking about, in principle, single trial activity of the network. And the larger part of, I think, what you're alluding to is actually the structure that you would see in mean activity. There is a little bit of analysis that they did on single trial as well, but a lot of what people think about in that paper is the mean activity. Having said that, I'm going to actually deal with a lot of mean activity here as well. Okay. Okay.
Sorry, is this projection something like what Sarah was saying from V1 to V2, for example?
No, we're going to be thinking about it within the same area as opposed to a projection out to another area. But both of these are relevant. So indeed, projections out to other areas. Well, I might allude to them at some point actually, because they are relevant for the motor cortex as well. Okay. So as I said, these data were collected by Dan and Werapong in Krishna's lab. They had animals doing this very standard center-out delayed reach task. So they touch a point on a screen. A little while later, a reach target, something that they're going to have to touch, is illuminated, but they've been trained not to reach immediately. They wait until a go cue arrives, often in this case, and often in other experiments, that the central point that they were touching is extinguished, and that tells them, okay, now go ahead, touch that point. If they do that, they get a reward of juice. So in these two animals, they had injected a viral construct which will basically infect the pyramidal cells due to this promoter with this excitatory opsin. So this is a protein which will express in the membrane, and when you shine light on it, it will depolarize the cell and make it fire. Okay? And the light is of a specific wavelength. Okay, so this means that you can turn on the light, and they do so through an optical fiber at around this, this is the wavelength they used. This is the power. So for people who are into this sort of thing, these numbers will become important, particularly in light of a very recent paper. But these were the parameters of the stimulation that they used. Okay, in this experiment, they found, somewhat to their surprise, that basically very little happened. There was no direct movement of the arm when they stimulated a whole bunch of cells in the motor cortex, contrary to what was known and is known when you stimulate electrically by passing current through the cortex. If they did so while the arm was actually in movement, there was no measurable deflection in the movement, so you neither initiated nor modified movements. There was a small effect in reaction time, so if you did this at quite the right time, the gap between that go cue being delivered and the initiation of the movement would increase by about 10 milliseconds, but that's 10 milliseconds in a gap of about 150 to 200 milliseconds, right? So we're talking about a 10% or less effect. Okay. So this was part of the puzzle. Is we didn't have much of this sort of effect. Now it does turn out in I think a recent I think it's still a bioRxiv paper, so pre-review, but it does look like if you turn on if you put in many more photons, you turn up the intensity substantially, then you can actually start to evoke movements even with optogenetic stimulation. And so one possibility is that, well, those parameters just weren't doing very much. But one of the nice things about optogenetic stimulation, as opposed to electrical stimulation, is that it doesn't interfere with your ability to record spikes electrically. And so they could measure this directly. And so this is, for a whole bunch of neurons that they measured, the change in firing rate with and without the laser, averaged over a whole number of tasks, as a function of the distance from the fiber where they're injecting the light. And you can see that so they actually had electrodes anchored onto the fiber itself. So right there, you got a range of perturbations, most of them excitatory, but a few suppression, in some cases a suppression of firing. And that pattern basically remains true at least out to sort of a couple of millimeters away. And certainly you see some influence even as far as 3 millimeters away from where the fiber is. Okay? So things were changing, and things were changing substantially, right? Some of these cells are changing their firing rate by about 150 hertz. You might also wonder whether 3 millimeters is a large area. This is a map of the motor, so both premotor and primary motor cortices. And this is sort of the scale of 3 millimeters in there. This sort of red, blue and purplish stuff is the general area that seems to be involved in arm movements. Okay, so these are the cells that modulate systematically when animals move their arms. And so, it's not the whole area by any means, but it's a fairly substantial area. And again, it's not inconsistent with the estimates that people have for the area that's invoked, where firing is evoked when you pass electrical current to create that. Okay, so another possibility was that, well, maybe the activity associated with controlling movements and executing movements is very structured. This is just a huge hammer. It's just kicking all of these cells up and firing substantially. And so maybe that direction of movement was just sort of direction of firing was somehow orthogonal to interesting directions. And this maybe comes back to this idea of communication subspaces. So there had been some work actually from Krishna's lab sort of 4 or 5 years ago, where they showed that you can interpret some directions in the space as being movement null. That is, perturbations along these directions don't correlate with changes in movement. So maybe we're just hitting one of these movement null directions in some sense. Okay, so one way to ask that question, although at this point not quite that precisely, was to say, well, rather than think about movement null, let's just think about the subspace that the task-related activity lies in. So just take the top principal components of the activity that we measure in these cells as the animal actually executes reaches. And we can now think about sort of not a movement null space here, but just an activity null space, which is just everything else, right? So the thing's orthogonal to that space. So possibly the impact of this laser, or of stimulating all of these pyramidal cells, would end up living in this null space. It would be orthogonal to the task activity space. So here are the time courses of the top 3 principal components as the animal reaches to the top right. And this is the time of the go cue, so there's a little bit of activity beforehand having to do with the preparation of the movement. In fact, most of the preparatory signal is in later principal components. The bulk of the variance, if you look at variance across the entire trial in these areas, is associated with the movement itself. The preparatory activity will show up in other principal components. They use different periods of stimulation. Here's one of the periods of stimulation they would use before the go cue. So this is during the delay period while the animal has the movement prepared. And here is the projection of the activity during stimulation into these same three principal components. And you can see that, in fact, there is a substantial projection into these principal components. And on the scale of the perturbation that's actually explored during the movement itself, it's quite large. Okay, so it's not that we have pure orthogonality. We'll come back to that point in a moment. And if you look at, just the stimulation perturbation is a vector in this recorded neuron space. We've got a subspace that we call task relevant and the orthogonal subspace to that. We can simply ask, take this vector, project into those two spaces, and sum the squared projections into each of those spaces. What's the proportion of the vector that falls in each of them? Well, more of it falls in the activity null space. The activity null space is a much higher dimensional space than the task relevant one. But the amount that falls into the task activity space is appreciable, as we can see here, and indeed is greater than you would expect if we just picked two random vectors or two random subspaces of that dimensionality. Okay, so there's actually a slight alignment between these two things.
Sorry, are you
Yes.
recording local field potentials, or are that the PCs of the local field
These are the principal components of spiking.
of spiking?
Yes,
Okay, so a rate.
a rate in effect. Yep. Okay, these are the other movement directions. So besides noting that there is a perturbation across all of the different movement directions, the other thing to note is that when the laser turns off, you get this very rapid return to, with a small perturbation, which lasts maybe around 100 milliseconds or maybe 150. You then sort of return and the system just continues to evolve as though there had been no perturbation at all.
Can I ask a quick
Yep.
clarification question, Maneesh? I was just wondering, so during the planning phase,
Yep.
does the optogenetic condition activity,
Mhm.
do they have different PCs essentially? Is it a
Yes,
completely different
yes.
subspace?
Well, most of the optogenetic input is indeed,
Right.
projecting into dimensions that are not. These are the PCs of the task. So there is a lot of projection into space, into a subspace that is not explored during the task itself.
And could it be that once you shut off the optogenetic
Mhm.
stimulation, the entire activity just returns to the aligned space where,
Well, that does seem to be what happens, but the issue is that somehow it's managing to erase this component of it that did project into this subspace as well.
I see. Right, got it.
Yes.
This reminds me of work from Svoboda lab, where they show attractor dynamics, where the optogenetic perturbation can kind of suppress activity, but then it kind of
Right.
becomes
So there's something. There's an weird asymmetry, at least from a theoretical standpoint, between excitation and suppression under optogenetic control. So in the work from the Svoboda lab, their observation was that it looked as though the contralateral field that they weren't perturbing was where the signal for restoration came from. Right? Now, if you have two sides of the brain and they have two different messages in them, you have to decide which one's right. And if you've suppressed activity on one side of the brain, then that's a natural signal to say, there's no signal there in effect, and therefore we just load over the signal that was on the other side. And that's what the circuit model that they built was in effect did. Here we're exciting the system, right? So in some sense you have to decide, these are spikes. These spikes are spurious, and some other spikes are valid. And the system, basically the question is, how does the system do it? So I think taking into account the other side of the brain, so we don't have measurements from the other side of the brain, and we don't know what's going on the other side. It might or might not be perturbed. There's reason to think it would be perturbed to some degree, because these spikes, clearly they're callosal connections, and so there will be some input over there. But in either case, even if we think of the system as a whole, we have to think about how you take a perturbed state and somehow erase the perturbation in order to continue successfully. So, whether we should be thinking about that as an attractor in some sense during the delay period, yes, it is an attractor, but you have a surface of attractors, so you have to decide which point on that attractor surface to come to. And in fact, the same thing's going to happen if the perturbation is delivered during the movement itself, at which time we don't have at least a static attractor at all. Yeah.
Is there any movement component that only PC1 can explain?
Is there any movement component?
So there's a movement
Yeah,
and that can be perpendicular to PC1. I mean,
So I wasn't really going to. This comes back to this issue of movement potent or movement null directions. Dan did do that analysis, and there is a significant projection into what we would think of as being the movement potent dimensions as well. That doesn't mean you couldn't build a decoder to ignore this if you wanted to, but you'd have to train the decoder to ignore this. It's not what the decoder would do naturally. So, Jim.
So here the pieces were determined by averaging all different directions of movement?
Yes.
I'm just curious. So I work with some colleagues who work with basal
Actually,
ganglia.
sorry, let me just clarify.
Oh, sorry.
Because I just realized you might have asked something. We didn't average all of, so it's the PCs of all of the movement directions, but each vector that enters into the sequence analysis is one movement direction at one time. So it's the ensemble of them. And then you have the principal components.
Variance between movement directions in the PC, so where do you see this?
You can, so it's subtle, right? But it's much of the variance is shared, particularly in the top three dimensions. But there are subtle changes between them. Sorry.
Just curious. These are well-rehearsed movements, and I work with some colleagues who work with basal ganglia and other subcortical circuits, and they would come and say that, well, motor cortex is now only initiating this, and this whole control is somewhere else, and therefore it's only.
So we'll come back to that, and that's a possibility that we simply won't be able to rule out. That's certainly possible in these situations.
But is there any causality established between motor patterns and the movement?
No. Again, I'll talk about that a little bit. Okay. So not only do we see little perturbation on the movement, but we also see little perturbation on the population as a whole, and that's really what we're going to focus on, rather than on the movement question per se. Okay, so there's some sort of robustness here, right? Somehow the system is able to erase this input. And so our question is, where does that come from? So maybe an extension of Arvind's question, right? Is the motor cortex actually involved in controlling the movement? And the answer really is we don't know for these sorts of rehearsed movements. Is that maybe the dynamics that we measure in motor cortex during the movement isn't even intrinsically generated, it's just being pulled in from somewhere else. Okay, so there's some other circuit, basal ganglia, cerebellum, somewhere else, where the dynamics are playing out, and we just see a shadow of those or a signal of those in the motor cortex. So that's possible, and as I say, I'm not going to be able to rule that out from these data. We just don't have the data to ask that question here. But it's an idea that I think a lot of motor cortical physiologists believe is unreasonable for the primate. So there's a huge difference as you look at different species in the nature of the motor cortical projection into the motor system. And in primates in particular, there's a very strong direct projection onto motor neurons in the spinal cord. And there's a lot of other sort of indeed somewhat circumstantial data supporting the idea that even in these movements, it looks like the motor cortex has an important role to play. But I think some more subtle hypotheses, like it's about initiation but not about control, or it's about initiation and then some sort of feedback correction mechanism but not about the ongoing generation of the control signal, still can't be ruled out. So, and I think that's a debate that a lot of the cortical community still have. Okay. But we wanted to at least explore the possibility that there may actually be something about the circuit which turns out to be robust to this sort of perturbation. And that's the direction that I'm going to take for this talk. Okay, and then at the end, sort of the new material is we'll try to ask why it is that this sort of specific robustness might emerge from a theoretical standpoint. Okay, so the first thing we did, and this was, so that result that I just talked about appeared at Cosine in 2014. A few years later, after scratching our heads about it with Leah, we ended up coming up with at least an understanding of how it could happen. But again, nothing that I'm going to say is going to be able to prove that this is the mechanism that gives rise to the result. And so what we did was to fit a linear dynamical system. We allowed for different degrees of time variation in the linear dynamics. So in some sense, it's really a nonlinear dynamical system, although that the nonlinearity or the change in dynamics was not fed back by the state. So it's sort of more a time-varying system than a strictly nonlinear one. And a noisy observation process. And we trained the system with a certain amount of information about the optogenetic input. So this, like a decoder that would be trained to reject the optogenetic perturbation, we were basically asking, can we build a dynamical system that can reproduce the observations that we see without having to change the dynamics to account for the optogenetic perturbation? So it has to be the same with or without perturbation. But while training, we knew about the perturbation vector. And the answer is yes, you can do that. Okay, so here are unstimulated and stimulated shards, as we've seen before. The structure that we're fitting is effectively a sort of a Kalman filter style model, which means there's a latent state behind it, and one can make inference about the latent state given the observations. And so these traces are basically the smoothed or inferred underlying latent states, but these are using the measured data to make the inference. So the data can overwhelm any intrinsic dynamics that the system would have. So then you can ask, well, what if I initialize the system at this location and now let it run forward without seeing the data at all? Are the intrinsic dynamics actually sufficient to create that pattern? And the answer is yes. So we can create the movement-related activity in response to when we initialize our dynamical system at that point. What happens if we initialize it there to the end state after the stimulation, and it does the right thing? So again, with indeed, interestingly enough, that little perturbation, it's actually successful in rejecting the fact that it started from this other location. So it's learned that there are directions in the space that should be reset. And by and large, it can get to the right place. Okay? It's not maybe perfect, but we're running forward.
Does the Kalman gain remain the same throughout?
No, because it's a time-varying linear system.
Because I see in PC1 on the left corner that there's a rapid gain. It looks like the gain is fairly high there. Whereas if you go to the third column, right to the bottom, so PC3, one of those, the one before, just this one, looks like there's and this actually matches the data. I noticed the same peculiarity in the data. So I was just wondering if there was some way you fit the gain?
Well, the common gain in this case would be a matrix. And so you would see different time scales on different projections just from a single common gain matrix. So I don't think the difference across the dimensions sort of comments on that. But indeed, the common gain is different at different points in time because it was a time-varying system. Yes?
So what is the dimensionality of the model? So it's the number of neurons in the data?
So we've tried a whole bunch of these things. So I think what I'm showing you actually is from a model with the dimensionality of where the dimensionality was equal to the number of neurons in the data. But I'm going to later show you results from a lower dimensional system, and it doesn't actually make a big difference in this case. So this system is reasonably low dimensional in the way it evolves.
Just to make sure I understood, you trained the linear dynamics on the standard data without the perturbation?
No, we showed it. So what we actually did in this case is we trained it on the dynamics without the perturbation, and then we showed it one of the perturbation epochs, and actually a very early one. And then it needed to know how to deal with that, and then we could test it on the other ones. Okay, so the model can learn to do this, right? And that's sort of where we were a little while ago. And so one question we might ask is, well, how does it do it? Right, so let's actually try to unpack this model. We've now got a model. Let's look into it and say, what is it doing? And in particular, how does it get around this conundrum of creating perturbations in these dimensions, right, which it does, and yet being immune to perturbations along those same dimensions? Right, that's sort of the puzzle that we wanted to get at. Okay, so, oops, I skipped over this. Right, so what does that, there's some state x, it's always latent, but latent and of the same dimensionality as the number of neurons recorded, or latent and lower dimensional. And there's some time-varying linear dynamics. And again, I've labeled this with a T in general, but it's sufficient in these low-dimensional systems, or lowish dimensional systems, just to have two, one before and one after the go cue. So basically, you need one for preparation and one for the actual sequence of movement. So let's take that matrix and do a singular value decomposition on it. So we'll break it up into an input space, a scaling, and an output. And now just in a little sort of cartoon way, let's imagine we're going to look at two of the singular vectors. So these are sort of input dimensions to the dynamics. We're going to project an input or a state onto V first before it goes through the rest of this matrix. Let's imagine that these are the output dimensions of the system. And then we can think about the scale factor, and we'll imagine that this pair, matching vectors V and U, have a low singular value, and this pair over here have a large one. Okay, so this kind of setup is sufficient to create the effect that we've seen, where you can get output variance, or at least you can reject dimensions that project along dimensions along which you have output variance. Let's put it that way. So the large singular value, so that's this pair over here, is going to tend to be associated with perturbations that are long-lasting in the system and the dimensions along which dynamics will be expressed. And you can see that if I have a stimulation vector set up this way, then it can have a projection onto the dimensions where that perturbation or where that dynamics is expressed, right, which is the output dimensions U1 and U2. So here we have equal projections onto both of these. But the projection onto the inputs is much different. Okay, there's a large projection onto this one and a small projection onto that one. And as a result, as we think about the action of this matrix A on our perturbation vector S, the first thing that happens is we project onto V, we then scale according to the singular values, and then we reconstruct by U, and now suddenly we have a much suppressed value. Right? So that's a very straightforward mechanism for how this would happen. If we had drawn eigenvectors instead of singular vectors, then in effect our eigenvectors, there wouldn't have been pairs, there would have been single ones, but they would not have been orthogonal. And so this is basically a phenomenon of non-normal dynamics. Okay. All right. So we could go back to the data and basically look to see whether something like this might be happening. So now we've estimated some matrices A. We've got a measured stimulus deflection or optogenetic stimulation deflection, which we'll call S. And we'll take that and we'll project it onto one of these matrices A. I think this is the average matrix A in this particular case. And indeed, we find that there's projection onto all of them. And if anything, there's sort of a stronger projection onto this leading dimension, and these have been ordered by singular values. So actually there's a large projection onto what's effectively PC1 in this space. Okay, if we now look at the corresponding input dimensions, however, we see that the distribution is actually does not have this peak over here. It's fairly even across these, but actually there's a little bit of a gap at the top. So there's very little projection onto one. And so now when we take this through and scale it out, we get an output vector, and that output vector still has some projection over here, but it's significantly reduced. Okay. And so now we can do the same thing, but look at neuron index as opposed to these singular vector indices, and just run through the whole matrix. And indeed, that stimulation vector is being suppressed successfully by our matrix A. But nonetheless, the sequence of matrices A is able to express variance along some of these dimensions where S does project into. Okay, so this was the solution to the puzzle. You can create variance in one dimension without necessarily having to be sensitive to that expressed variance. Okay, and so we can think of this as a sort of a dynamics null space. We had communication null spaces or communication spaces in the work that Sarah pointed to. As I mentioned in thinking about motor cortex, people have thought about movement null spaces, so spaces in which you can have activity during planning without executing a movement. And this suggests that you might also have sort of dynamics null spaces where you can either have input perturbations or indeed perturbations expressed through the dynamics that don't have longer-term dynamical consequences.
Have you looked at how these either input or output dimensions compare to the principal components? You showed us earlier the top three principal components of the linear dynamics.
Yeah, so the output dimension is strongly correlated or is close to the leading principal component, because those are the dimensions that are expressing the variance in the output.
So you need some input to come in. So I guess how low down do you go in the eigenspectrum before you find the principal components? If the system was really only three-dimensional,
Yep.
three principal components, and those were also the output dimensions, and the input dimensions were orthogonal to them, then you have no inputs.
If it was strictly three-dimensional, right? But in fact, at each time you're sort of distributing across, exactly. And a lot of the dynamics is being carried by relatively low variance signals.
Okay.
I mean, that's essentially what it's saying. And again, I should just stress, it's a solution, right? It's a way to sort of put together these two observations by training a model to do it. Okay. Right. Okay. So the next question is why, right? So we've trained a model to try to do this. It can, but is there any good reason to expect that the actual dynamics should have this sort of structure? And there's sort of a natural thing to look at, which is the nature of the stimulation here. It's not a sort of random direction in the space. It's kicking a whole load of the excitatory neurons substantially, right? Not all of them, because there's viral infection issues and there's also a local fiber. But we're basically pushing in a somewhat preferred direction within the space. And a natural reason for a large perturbation along this dimension to be lost would basically emerge from having this perturbation embedded in some sort of network that has some balance between the excitation and inhibition within the network, and we'll come on to that in a moment. But this led us to sort of a problem as data analysts, which is a lot of our work involves sort of abstract networks of neurons. They're sort of sometimes barely networks. They're just sort of linear dynamical systems. And these sorts of statistical models, which we could draw as saying we've got some latent variables evolving in time, we have some recorded data, and we assume there's some distributional dependence of the recorded data on those latent variables. Right? They can describe the data quite well. They capture a lot of variance in the data. They allow us to perform this sort of parametric fit and then analyze it. So they're good ways to extract. Sometimes I think of these as sort of the modern version of the PSTH, if you like. They're just taking our data, reducing it into a way that we can then use some intuition to try to understand what's going on. But it's quite hard to actually take one of these as it stands and engineer in things like biological constraints, such as our EI, our desire to try to understand whether this could happen through a combination of excitation and inhibition. Okay. What we'd like to do is we'd like to be able to take a network something like this. Now these of course have been studied extensively, but often in the context of either a simulation or mean field approximation, mechanistic model, or the mean field equations will often lead you to writing down something in continuous variables like this. And so these are just sort of the Wilson-Cowan system over here. Okay. But we can still, it's easier to engineer into these the biological constraints we want. We can have separate excitatory and inhibitory populations straightforwardly. People have designed such networks to achieve various sorts of computation, right? So with a certain amount of crafting. And you can also, when you do that, often set things up, usually by a lot of exploration by hand. You can find ways in which a network will create phenomena which will match certain selected features of the data. But in general, though, you've just picked the dimensions you want to align things with, right, in some sense. If you ask how much of the variance overall in the data are explained, it may not be a huge amount. And so what we wanted to do was to try to think of ways that we can basically merge those two things together. Okay, so let's just sort of think a little bit about the state space modeling kind of view before we go there. So again, we have some latent variables, they evolve in time. We assume that evolution is what explains the temporal evolution of our recorded data. And so our data live in some high dimensional neural space where we measured them, and the dynamics are usually encapsulated by some sort of evolution in a low dimensional space, maybe because there's a low dimensional task involved. And so we try to identify that space. And even though we haven't recorded all of the neurons involved in the circuit, we hope to be able to recover the essence of this low-dimensional task dynamics, having used some sort of incomplete sample of the full network. Right? That's sort of the framework that many of the talks you've seen have been working in. Okay, so writing this out, in this case with a time invariant system, we have our latent variable evolving, in this case linearly, with some sort of injected noise that we often think of as innovations. It both allows for some variability trial to trial and also allows us to deal with model mismatch. So it's a term that we can sweep up any nonlinearities in the system into an estimate of that noise. Our observations are taken in this case again to be linear dependent on x, although you've seen models where that linearity can be relaxed. And again, we assume that what we actually measure is some perturbed version of that. Okay, so how do we put circuit constraints into this framework? So what sorts of constraints might we be interested in? Well, we might be interested, first of all, in having some network that actually satisfies Dale's law. We might like for there to be some sort of balanced connectivity within that. We might also actually want to extend this to thinking about spatial connectivity, right? So that things that are nearby are more likely to be connected than things that are far apart. And there's many other constraints you could imagine. Another sort of slightly subtler constraint, which comes up when you start to think about these systems, is that it ought to be the case that our recorded data are a random sample of the elements that would sit within the circuit. And that actually, I think, is quite a strong constraint, because in some ways you can fit a system like this to explain your latent data, your recorded data, but often when you do that, what your high dimensional system of your sort of biologically plausible network is doing, it might have lots of behavior for which the things you recorded are atypical. And that is unlikely to be valid, right? It's much more likely that what we recorded here were just some typical examples of what was going on in here. And that's a constraint that we don't quite know how to enforce mathematically here, but it will kind of come back in our thinking about the validity or otherwise of some of the solutions that we turn up. Okay. At the same time, right, we'd like to have these constraints from the circuit. We'd also like to have constraints that would emerge from the recorded systematic data during the experiment. So indeed, somehow the system should be consistent with the fact that what we actually observe looks like it's relatively low dimensional, right? So if we're going to learn something that lives at the circuit level, it had better express low dimensional task activity. And it should capture a lot of the variance in the firing rates that we actually measure. Okay. So the way we went about this was to introduce a third system, and all of these you can think of as this one hangs off the latent system X. This one, in some sense, runs in parallel to the latent system X. So this is evolving in time, and the latent system X is also evolving in time. W, the connectivity matrix here, is the place that we will actually impose all of these constraints, right? So these things, the requirement for Dale's law is a sign constraint on W. So I should be a little careful here. We're going to constrain the signs of W. We're not going to constrain nu. Okay, so there's the firing rates. So I can have negative firing rates in an inhibitory cell, which of course will make it excitatory. So there's something a little funny about the way that this is implemented at the moment. That's because we're still trying to live in a sort of linear domain here. But one way to think about that might be that nu actually is reflecting perturbations from some sort of background rate of firing, and then indeed suppression would be fine. Okay. The constraints from the data. One way to think about what they're telling us is that this matrix A that we've identified, or the sequence of matrices A that we've identified from our latent variable system, should somehow be implemented by a projection of this high-dimensional system. And so one way to perhaps try to get this idea of random sampling into this is to pick a random projection J and insist that the evolution of the low-dimensional system should be implemented by taking the pseudo-inverse of J, projecting it up into the high-dimensional space, so projecting X into the high-dimensional space, running through the network dynamics you have over here, and then projecting back down. Right, so we can try to tie these two systems together essentially by this relationship. And this is sort of saying we want this to be as close as possible in a mean-squared error sense. That's where the pseudo-inverse comes from. Yes.
I think you might have mentioned it before, but I forgot what was the reason why you did not want to include feedback into the network?
feedback into the network. Sorry.
So it's like a minus new term, so like a state dependent essentially.
Not sure I quite understand. So this is a vector. We're just in discrete time. So there's diagonal element. So diagonal entries in W are self-feedback. Yes.
I'm curious, why do you need to enforce low-dimensional dynamics if data is low-dimensional, and you just have this network, and you project directly from new to Y spot simulations? Would news and be like automatically low-dimensional?
So there's no particular reason why it should, right? It can do a lot of, it can basically, because this W can be, essentially it can use a lot of non-normality to create useful dynamics in the expressed output space. If I've applied C directly to W, then I can get a very high dimensional activity here such that the effective such that the Y still follows the recorded trajectories, but our high dimensional system is not. And this is where this slightly subtler point about the fact that Y should be a random sample of nu comes in, right? So just empirically, I mean, there's no theoretical reason why it should, and empirically we find when we fit these systems they don't. Basically, the constraints that you've put on here make solutions that allow it to be higher dimensional much more likely from random initial conditions.
Sorry, I just
Sorry.
trying to understand what you just
Yeah.
said.
Okay.
So
It's a crucial point. So this is.
does this relate to the sampling issue? Because I mean, if VT has the same dimension as YT, and you want to fit VT to YT, why wouldn't you expect to have it the same dynamical dimensionality then?
So if nu t and yt were of the same dimension, then basically we would just be trying to fit something where our measured neurons, we were trying to fit a model where our measured neurons all depended on themselves, in effect, right? We had a one-to-one mapping. And then we would just be allowing some sort of rotation between these things. And so in that situation, yes, you would need to find a low-dimensional system. But you can't, that's actually that constraint, we know that the neurons that we've measured are embedded in a much higher dimensional network. In fact, given these constraints, it's quite hard to find a system that has just the same number of measured neurons that will do the same thing, right? And so when you allow nu to be much higher dimensional, now it will tend to express more dimensions than you've measured in your Y.
I'm curious, why do you need to run dynamics on X's? Why couldn't you just project nu on some low-dimensional?
So in effect, that's what ends up happening. In this scheme, we've used X to extract A first, and then we're constraining W through this. But it looks like you don't need to do that. And so I think, in fact, the data that Leia's given me to show you, the actual model fits, are from a later iteration where it's still using the random projection J. So we're still looking for that, picking a random direction out of nu space, and then learning C to, so I should put this on here, then learning C to act on J times nu, but the X is actually dropped out. Yes?
I'm a little bit confused about what W represents in the network variable.
Yep.
So in the continuous dynamics, effectively that's discretized, so
Yep.
I imagine that the W is exponential of some weight matrix times delta T, isn't it? Linear dynamics, so
Yeah,
decay factor is
there's an interesting question as to when you extract a spiking system to continuous time, kind of where did the weight matrix go in the first step, right? And then when you now discretize, where does the weight matrix go in the second step, right? So it's actually quite, like the differential equation form is itself not actually a good model of the underlying system when you're actually thinking about neurons, because it's spiking.
a bit confused. Which one should follow the Dale's law?
So one way to think about it is if I'm just binning spike counts in my system and building a discrete system that way, then the spike count should, the spike count to spike count influence should follow Dale's law, and that's the W that I have here. Did that answer your, right? So I've imagine that this is a count, right? I did not touch anything this time. Here, I'll put this away. So if that's a count, right, then the total number of spikes in an inhibitory cell in the preceding time frame should reduce the number of spikes in its targets on the next time frame. So W should be negative.
Yeah, so it's not negative, right? I mean, it's just
There's a time.
decay factor.
Well, the influence should be negative, right? W is going to, that's now I've got a linear combination of all of the influences, but the influence of increasing the number of spikes in one should, yes, there's a time constant in there, so W is not actually the synaptic weight.
Exactly, so that's what, I think that's what Darrell was saying, that W is the integrated direction.
But I think if you try to go, if you write down Dale's law on the continuous time system, right, then now you've got e to the, as he said, you've got this exponentiation of things. So, but it's kind of weird to have Dale's law in the continuous time system, right? It's that leap which is the weirder leap, I think. We can come back to this maybe.
Sorry, so just one
Yeah.
question. So the CJ, so on the second level, so why? So,
Yeah.
So does it mix and different news?
Yes, both J and C are mixing things.
Because I would think that the recorded data is some subsampling of news, but here you
That's
said
right.
it's
No,
mixing
so we're
of
not.
news.
So we're not trying to implement it as saying there is a subset of the news that we have actually recorded, although that's an underlying notion, right? We are projecting through the low-dimensional space, and then projecting from the low-dimensional space to our measured Ys. But the dimensionality here is still going to be a few hundred, right? It's going to be far short of the millions that the area actually has. All right. So, so pretty much out of time actually. So the model. So we can fit a model like this. It is capable of recreating, this is the unstimulated perturbation, so we can get these long time scale, this sort of long time scale evolution out of a system that looks like this. What about now injecting optogenetic stimulation, right? So what happens if we now inject something that is going to, and the way we'll implement it is, now that we have an EI network, we're actually just going to drive all of the E cells in our EI network. So what happens when we do that? Well, again, the intuition behind building this was that we should expect those sorts of dimensions to be eliminated. So suppose we just chose our W's each individually IID from some distribution that just depended on whether we were looking at an excitatory or an inhibitory cell, right? So we just have a matrix where there's no structure in here besides this, just one set is excitatory, one is inhibitory. So then if we look at the eigenspectrum, we're just going to get this relative, depending on our choice of this distribution, as long as our amplitudes are not too large, we'll just get something that is a disk of eigenvalues. And now if we inject, say, this dimension S, then the most, it will decay at the slowest rate at which it will decay is set by the kind of largest eigenvalues in this space, or the largest absolute values of the eigenvalues here. But in fact, this particular axis, which hits all of the excitatory cells, might decay much faster, and a lot of people have observed this about EI systems, because if we have, so these came in the wrong order, if it's the case that each cell has on average, because of this independent structure, roughly equal input from all of the excitatory cells, then as we pass this input through here, we'll just get sort of roughly equal activation. And now if we take that and put it through a balanced network, we get no output. So in discrete time, in 2 time steps, we should expect to be able to get rid of this thing. Yes?
So what do you mean by balanced?
What do I mean by balanced? No. So here I mean that the net excitatory weight and inhibitory weight, so by balance I basically mean exactly this, right? I mean that if I stimulate all cells equally, the net current onto every other cell is 0. So tight, very tight balance. Very tight. So in this case, precisely tight balance.
Detailed as well. Like every row sum is 0.
Yes, every row sum is 0. That's what I mean.
More generally, people look at inhibitor-dominated networks, and then you get one node that sticks out on the left, and
Which is actually this,
exactly.
yeah, which is this decay mode, right? Yep.
So you don't have an eigenvalue that is sticking to the left node?
So again, those are the continuous time portraits, right? So these are discrete time portraits. So here 0 is the fast decay as opposed to negative infinity, right? Because these are not exponentiated, or these are exponentiated, right? Because they're discrete time eigenvalues. Okay, let me just try to very quickly show. Okay, so this is the random case. What we have is a network that was trained to create a particular pattern of activity, right? And so, in fact, if you look at the eigenvalues necessary to create those low-dimensional task dynamics, they have this pattern over here, right? So there's some that lie very close to the unit circle, and those are the ones that are carrying the slow dynamics that are necessary for creating the movement. And so when we train this network and look at its structure, we actually get something where, all right, wrong order. But the blacks actually do lie on top of these red ones. There's the same eigenvalues do appear. They appear so perfectly that you can't see them because they're behind the reds. I'm sorry, they're plotted in the wrong order. Okay, so we can create these task activity patterns, and we can see the signature of it in the eigenspectrum as well. And so that but this sort of pool of random eigenvalues in the background still exists. So there's still this kind of, in the task null space, we haven't perturbed things, and we still have that set of extra eigenvalues over there. Okay. And so now what we can do is we can actually do the thing that Jonathan was asking whether we had done at the beginning. So now we can train only on the non-stimulated data, and then evaluate what happens with the stimulation. And the idea is that this EI structure and basically the enforced tight balance is what will get rid of the stimulation vector. And so that's what we find here. So this, again, are the data, unperturbed. These are the various different perturbations. And we're now seeing all the different stimulation periods, the 3 leading principal components, and the different conditions, so the different reach directions in the different colors. So here, I guess you can see, for whoever was asking about the variance across reach conditions, it's perhaps clearer here because we plotted them on the same axes. And this is what you've already seen. We can reproduce that. And here is the reproduction of now the perturbation due to this common excitation of this excitatory cells in the network, and then the sort of, I guess in this case, because you don't see the non-stimulated and stimulated on the same plots, we have to compare across the 2 lines. But essentially, we've been able to recreate these perturbations. I'm trying to point to one of these. Let's see. I'm actually looking for. Right, so if you look at the profile from the offset here, you both see this slight overshoot in the negative side, which then recovers back to this point, which is absent in the unstimulated case. And that sort of post-stimulation slow decay has actually been captured by the network. And that's actually coming from this disk of the other small eigenvalues in the system. So those are important to reproduce these sorts of decays. But also, subsequent to that, you get the same dynamics being played out to execute the movement. And the same is true in the other dimensions. Okay, so let me finish because I'm already over time. So, so when you kick these cells in PMD or M1, you have a very strong immediate impact on neuronal firing. We can see that directly, but very little impact on behavior and little impact on the future evolution of dynamics in the neural system. And this is true even though the perturbation that we measure in the neuronal firing has a substantial projection into this sort of task manifold, which is just PCA on the data. Okay, so one possible interpretation of what's going on, but not, well, I guess this in itself is just a restatement that this perturbation is dynamics null because it doesn't affect the dynamics. But one way for that to happen is that it's dynamics null within the circuit that we're looking at directly. as opposed to sort of the trivial form of dynamics null, which is all of your dynamics are coming from another circuit entirely. Why might we have so that's possible. We can reproduce the data under that hypothesis. And one reason that we might imagine that would be true is that it may be that this sort of robustness to common excitation is important effectively for stability. And that common excitation would be either of all neurons or of all pyramidal cells. And it might emerge from the balance, and it may help to separate sort of the fine dynamics that are necessary for the computation from what we might think of as gross population dynamics, such as the beta traveling waves that we know exist across these areas. Okay, let me thank everybody again, and then I'll take more questions. Yes?
So if I can try to restate,
Yep.
What you're saying is that basically by tuning the network,
Uh-huh.
it generates dynamics in a subspace
Yep.
that has a significant projection onto the uniform, but
Yep.
it's not the uniform, because critically
Yep.
the uniform is quashed by the EI dynamics,
Mm-hmm.
whereas something,
Yeah.
whereas the
But in
task,
a linear system,
yeah.
having a projection onto that dimension would have.
So I can see how from
Right.
kind of more as a normative, like if you think why would it be set up that way, I
Yep.
can understand your last argument, why
Yep.
you would want to squash. What I don't fully understand is why is the other
Why do
one
you
loading
why
instead
do
of
you
making
create
it
variance in
completely
that?
orthogonal?
Yep. Okay. So I think that's an excellent question. It's one that we don't have a perfect answer to. We only have speculations to. Right. It's one that we've worried about a lot. So I think so one explanation is that, again, so once you set things up with the Dale's law, you're inevitably non-normal. It looks as though a lot of, so you can write down the surety composition, for example, for one of these things.
Mm-hmm.
And depending on how you order the eigenvalues, you'll often, well, so surety compositions are not unique, but if there's, I guess for many of the systems that we'll write down, you're often getting loading into this rapidly decaying dimension. And it doesn't cost you anything because it's rapidly decaying, right? So I don't have a good answer, right? I mean, it's something which, as we played around with these things, is emerging from the EI networks when we try to fit them here.
So you're saying it
But
doesn't hurt, and that's why.
And so then there's no penalty
there's
to
no
having it
pressure
there.
for
There's
putting
no
it there
pressure
because it
to
doesn't
get
hurt.
rid of it. So it emerges somewhat naturally, and there's nothing quenching it. But that, as I admit, it's not a great answer.
So I'm confused with something. So after movement initiation, so you have this big component on the uniform mode.
Yes.
So how is this generated if the network is quotient?
So again, so the network is not sensitive to inputs along that dimension,
Okay,
but
so
does generate outputs. So it's just the non-normality of the dynamics.
but in your
so-
model, how do you generate it without an input? Actually?
Well, inputs in other dimensions are generating it. Sorry, so, okay, maybe there's two different questions. So how is it that we change the dynamics from preparatory phase to movement phase? That is, how does the go queue appear in the system? Okay, so there's at least two ways to do it, and they are basically equivalent in the models. One way is that we actually change the dynamics in the low dimensional state at that time. In this network, so in the EI network, didn't make a lot of sense to change the connectivity suddenly. And so in this network, there's basically a single sustained condition-specific input during planning, and it switches to a different but sustained condition-specific input during movement. So at the go queue, there's just a switch of what the input is, but the input is just fixed over that time. So the time course doesn't come from that. It's just a push in one direction, which is, in effect, it was able to implement the change in dynamics by just shifting to a different location in that space. Okay. That's fine by me.